Okay, folks, let's get started. As you can see on the screen, I do not have grades posted. I will have an announcement about the exam at the end of the lecture today, however. So that'll keep you tantalized. I spent some time last time going through most of the mechanisms, and I finished talking a little bit about restriction endonucleases, or restriction enzymes, which we also call them. And I want to just say a few words about those, and then we're going to move on to catalytic regulation and control mechanisms. So as I indicated last time, restriction enzymes are similar in mechanism to what we saw with proteases. We remember, of course, that restriction endonucleases are cutting DNA, not protein. And DNA, of course, has nucleotide building blocks, not amino acid building blocks. And further, we remember that the bonds between nucleotides are phosphodiester bonds, not peptide bonds. So there are some different terms, there are some different things there, but the similarities are that we're using an activated water molecule as a nucleophile to break phosphodiester bonds, and that's a very important consideration. Students frequently find interesting the sort of story of restriction enzymes, so I'll spend a couple minutes talking about that. Restriction enzymes are, as I said, protein or enzymes that specifically cut DNA at specific places, and you might wonder why such enzymes exist, because we think of the genome as being something that we want to protect, we don't want to damage, et cetera, et cetera. And it turns out that restriction enzymes are produced in bacteria. They're not produced in human beings. And bacteria use them as a sort of primitive immune system. It's a very simple, primitive immune system. In the immune system, we have antibodies that attack things that are recognized as foreign. In bacteria, bacteria get infected by viruses just as we get infected by viruses. The viruses that infect bacteria are known as bacteriophages. And these bacteriophages typically have a genome of DNA, very much like other viruses have genomes of DNA. They can have genomes of RNA, but most of them have DNA. And part of the infectious cycle of any virus is that the virus must attach to a cell and inject its nucleic acid into the cell. So in the case of a bacterium that gets infected by a bacteriophage, the bacteriophage has grabbed a hold of the cell, and it will inject its DNA, if it's a DNA virus, into the bacterial cell. The bacteria make restriction endonucleases as a protection. So they recognize and cut specific sequences, and if that invading virus has those sequences, which it typically will because the recognition sequences are relatively short, typically 4 to 6 nucleotides in length, then the invader will basically get chopped to bits. And one of the questions that arises then is, well, why doesn't the bacterial DNA get chopped to bits as well? And that's the last part of the story I need to tell you about restriction enzymes. Restriction enzymes are part of a system that we refer to as restriction/modification. And the restriction part is the enzyme that does the cutting, as I described to you. The modification system I haven't told you about. And the modification system is comprised of another enzyme that recognizes exactly the same sequence that the restriction enzyme recognizes. That is, if I have something that's an EcoR5, then that EcoR5 restriction modification system will have a second enzyme that recognizes GAT-ATC, just like the restriction enzyme that does the cutting does that. Well, what does the second enzyme do? The second enzyme doesn't cut that sequence. Instead, it puts a methyl group in the middle of that sequence, a methyl group. All right? So the modification enzyme does that. It's called a methylase. And when we examine that, we see in the case of this particular sequence, there's the unmodified enzyme sequence there. There is the modified sequence, and we see that a methyl group has been put onto the A of the GAT-ATC. That means that one will go on the bottom A, obviously, as well. And the significance of that is that single methyl group prevents water from binding in the right place to make the cleavage. So even though the enzyme can recognize that sequence, water can't get in there and do the attack and cause the bond to be broken. So when a DNA has been modified by the methylase, it will no longer be cut by that same restriction enzyme that recognizes that sequence. So, for example, this guy has been modified. This could be in the cellular genome, and it's protected from cleavage by the restriction enzyme. The question then arises, well, can the methylase get to the invader phage before the restriction enzyme does? And the answer is yes, it can. And if that happens, then the phage will survive the being cut and go on and infect the cell. You say, well, why does that happen? Well, it happens because there's no perfect system. Your immune system is not perfect. The bacterial system is not perfect. But suffice it to say, this protection system that bacteria have is pretty darn good. If you've ever worked in a laboratory and you've tried to put plasmid DNA sequences into a bacterium that has a restriction modification system, you find it's very inefficient to do. Most of the things you try to put in get chopped up, and you don't get any plasmid in at all. But at a very low frequency, you do get some plasmids in, and then they get methylated regularly and survive that process. Okay, so that's the restriction modification system. As I said, it exists only in bacteria. It does not exist in human beings. And we don't need such a system because we have an immune system that, in theory, is protecting us from invaders as much as possible. Okay, questions about that? Yeah, back there.
Is it possible for the modification enzyme to mistakenly go onto the virus genome?
Yeah, so that was the question I was saying. Is it possible that it'll get there before the cutting enzyme can? And the answer is yes, it can. So if that happens, then the invader will actually be protected. It will replicate and it'll kill the cell. So as I said, it's not a perfect system, but it's a pretty darn good system. Shannon?
Did you just say that we don't have modification?
We don't have restriction or modification. So
we
have neither.
Okay.
Yeah. And again, we have an immune system that's protecting us extracellularly, not intracellularly. Yeah?
What happens to the methyl group after the invader is chopped off?
What happens to the methyl group?
Yeah, after the enzyme nowadays thing.
After what?
After the enzyme kills the invader.
After the enzyme kills the invader. Oh, I see. So once if the enzyme kills the invader, then that means that it didn't get methylated. Right? If it gets methylated, then it's just going to stay on there. It's not going to do anything. It's just going to be protected. Okay? Yes, sir.
Doesn't that methylation enzyme also get assistance from the actual, during the reproduction of the DNA itself, when there'll be another reader that comes along and matches methylation stage from the parent strand to the new strand?
I'm sorry, say it again now.
If you're copying the DNA,
Uh-huh.
does that methyl get copied as well?
Okay, if you're copying the DNA, the methyl will get copied as well. The methylase still has to come in and do its thing. So is it possible that the restriction enzyme may cut that at a low frequency? And again, the answer is it's possible, yes. So the methyl doesn't get copied. The methyl has to be put on after the DNA is made. Connie?
When you say we don't have a restriction modification system, does that mean we don't have any restriction enzymes or modification enzymes at all?
We have no restriction enzymes, no modification enzymes at all. That's correct. And I'm not sure if I answered your question properly back over here, so let me say one more time. If the methyl group gets put on, then the invader will replicate and kill the cell, basically, okay? Because it won't be able to be cut by the enzymes. So if that happens, then you just have plenty of viral DNA. The methyl group doesn't do anything at all, because as far as the rest of the proteins of the cell are concerned, it's just a GATATC sequence, okay? If we're talking about how we recycle nucleotides, we'll talk about that a little bit when we talk about nucleotide metabolism next term. So I'll save that for that point. I hope that better answers the question for you. Okay?
Are these all found in like the nucleus?
Well, bacteria don't have nucleus. Yeah. Okay. The one last thing I want to say, and this doesn't really relate to mechanism so much, but it does remind us of the importance of shape changes in proteins. So the last group of proteins that are of considerable interest with respect to catalysis and so forth are known as the myosins. And myosins are part of the actin-myosin pair. And actin is one protein, myosin being another. And these proteins are very, very important for, in fact, they're essential for muscular contraction. So these these proteins together produce the contraction that occurs inside of muscles. All right. Now, there's a whole bunch of stuff with respect to mechanism, and I just don't think it really tells us much about mechanism that we haven't already seen. No surprise, you're going to see an activated intermediate that's going to cleave ATP. But the point that I want to make about actin and myosin is this. Contraction happens because myosin literally crawls along the actin. And that crawling requires molecular change in a protein, this protein being myosin. And I want you to look and see this protein. Here is myosin. And when ATP, in fact, what this protein does is it hydrolyzes ATP. And that hydrolysis of ATP induces a significant shape change in this molecule. You can see that the unhydrolyzed ATP, and after ATP hydrolysis has happened, this motion has happened inside of this protein. This motion, folks, is what allows you to move. It allows you to have a heart. It allows you to have all kinds of motion necessary to function. And it's happening because of the shape change of a protein. This motion right here we could think of as like a claw that is allowing the myosin to crawl its way along an actin filament. Okay? A really cool thing. And that happens as a result of this shape change in this protein. The shape change requires the hydrolysis of ATP. So a very cool application of shape changes that we see in proteins happening as a result of catalysis. Most of the rest of the protein, you'll notice, doesn't really change much. It's only this section out here. A picture of myosin is actually on here. You can see these two little heads that are out here, and these heads actually crawl their way along an actin filament. Okay, that's what I want to say about mechanisms of catalysis. With that said, I would like to turn our attention to discussing the allostery and regulation. So allostery and regulation, or control as I often at times describe it, is very, very important with respect to the needs of a cell to get a harness on enzymes. Now, earlier in the term, I mentioned that enzymes can be extraordinarily efficient, extraordinarily fast, and I gave the analogy of driving a Maserati to Fred Meyer, at 110 miles an hour. You could imagine there's going to be some problems with that if you don't regulate in some way. We regulate with speed limits. Cells regulate enzymes by a variety of mechanisms. So one of these mechanisms is known as allostery. And I've mentioned it briefly before, but I will say it again and also give you the definition again. And allostery, or allosterism, is the mechanism by which the binding of a small molecule to an enzyme affects the enzyme's activity. So binding of a small molecule to an enzyme that affects the enzyme's activity. Now, when I mentioned this before, I pointed out that not all enzymes are regulated. Cells are very efficient in regulating things. They regulate the most important, or say the first enzyme in a metabolic pathway, and by controlling the first enzyme, they control all of the things that flow through it. Just like if I put a toll booth out in front of I-5 in Albany, and I stop all the cars there, there aren't going to be many cars getting through, only the ones that the toll booth allows through, right? So it's the same thing that happens in metabolic pathways. If we control that first enzyme that catalyzes the first reaction in the pathway, we can essentially control the whole pathway very easily. It's very efficient, and so that's a very useful thing. There are in the cell three main mechanisms that cells use to control enzymes. One of them is allosterism, what I've already described to you. I'm going to show you some details of allosterism later today. A second modification, or a second control means that cells have over enzymes is covalent modification. They can covalently modify enzymes. We'll see some examples later which involve the addition or removal of phosphates from enzymes. And these covalent modifications can activate or inactivate enzymes depending upon the enzyme. Allosteric can be positive or negative, that's correct. The third mechanism that cells use to control enzymes is controlling whether or not they're synthesized. That is, whether or not the protein is even made. And that seems like, well, duh. It turns out that's one of the most important considerations for many control systems. Is the protein being made by the cell or not? And that control is exhibited in several ways. It can be transcriptional, it can be translational, and we'll talk about some of those next term. Okay. So what I want to do now is talk for a bit about allostery and this very interesting enzyme called ATCase. So let me show you a little bit of this. ATCase catalyzes a reaction that, to be honest with you, we're not going to pay much attention to the reaction itself until next term. But it catalyzes a reaction that is a very, very important reaction because it's the first step in making pyrimidine nucleotides. The first step, that is the very first reaction in making a pyrimidine nucleotide, is catalyzed by the enzyme ATCase. Now, the enzyme ATCase has a much longer name. It's known as aspartate transcarbamylase. I'm not going to spell that here. You can get it out of the book if you want to get the word. We commonly abbreviate it ATCase, but please note that when you use an abbreviation, you have to get it right. You can't call it ACTase, for example. ATCase is the proper name. Okay. Now, here's the reaction that it catalyzes. You can see it on the screen, and it involves an aspartic acid. And one of the things that we'll see that's of interest next term is that all of the nucleotides that are made by cells have building blocks that start with amino acids. So we can start with very simple things and make fairly complex molecules like nucleotides using various enzymatic systems. Now, the pyrimidine nucleotides include CTP, UTP, and TTP if we're talking about DNA. Okay? To make these nucleotides, it's more than one reaction. And this is why I refer to these things as pathways. To go from these simple molecules here all the way down to the final product, which in this case is CTP, takes about 10 steps. About 10 different reactions are necessary. Yes? What are the pyrimidine nucleotides? That would be CTP, UTP, and TTP. Okay. But it takes about 10 steps to get to this final product here. Well, as I said, cells have efficient means of controlling pathways, and cells really don't want to make too much of any given nucleotide. We know from the study of cells that have aberrations in them that cause them to have too much or too little of a given nucleotide cause those cells to have higher rates of mutation. Mutation is generally not a good career move for cells, certainly not in the short term. Over evolutionary history, yes, but over the short term, most mutations are very detrimental to cells. So cells are very much what I describe as control freaks. They put a lot of energy and a lot of controls in the way of preventing the nucleotides from getting too high or too low. Okay? Well, what does this all mean? Well, let's imagine that I'm a cell, I'm sitting around, and I'm making pyrimidine nucleotides. I produce CTP, and the CTP concentration starts to increase. But if the CTP concentration starts to increase too much, I don't want to make any more CTP, so I want to turn off the synthesis. I want to turn off that pathway. And it turns out that the enzyme ATCase ATCase is an allosteric enzyme, and it will bind to the end product of this pathway. So ATCase will bind to CTP, and that's what this little red thing is showing here. And when it binds to CTP, the enzyme is turned off. So when the enzyme binds to the end product of the pathway, the CTP accumulation starts to get high, the enzyme gets turned off, then that shuts off essentially all the reactions leading up to CTP. And until that CTP starts getting used, that pathway will essentially be turned off. Now notice I said essentially. If you remember, I said we think of these mechanisms in terms of on and off, but in reality they're more like turning the volume down or turning the volume higher, but we still have some things coming through. Okay? So allosteric mechanisms do not have on-off switches, but they have turn-down, turn-up situations. Well, the beauty of this is the end product helps control itself. That's a very, control its own synthesis through this enzyme. It's a very, very useful thing. It's a very simple mechanism. And as a result, CTP concentrations inside of cells don't get too high. You say, well, what about UTP? What about TTP? Well, it turns out UTP and TTP are both ultimately made from CTP. So by controlling CTP, you're controlling all of the pyrimidine nucleotides. One step, one enzyme, one molecule. It doesn't get any more efficient than that. So that's really cool how cells are able to do that. This mechanism I've just described to you has a name. It's called feedback inhibition. So feedback inhibition occurs when the end product of a pathway inhibits the first enzyme in the pathway. Feedback inhibition occurs when the end product of a pathway inhibits the first enzyme in that pathway. We'll see other examples of feedback inhibition, primarily next term, but there are many examples that cells use of feedback inhibition because it is so simple and easy to control things. Okay. Well, that's one interesting aspect of ATCase. There's the actual reaction that is catalyzed, and no, you don't need to know all this stuff that's on here. I'm just showing you. There's the carbamyl phosphate. There's aspartic acid. There's the intermediate that it makes. There's a whole bunch of steps. And there's the final product of CTP right there. Now, I find it really cool and remarkable that the nucleotides can be made from amino acids. We know that amino acids exist in space. We know that amino acids can combine in space, and people have actually found nucleotide precursors in meteorites floating out there. So this idea of the chemical evolution of life is pretty cool, and we use the materials that are available to us to make us and make life possible. Okay. Well, if I study this enzyme, ATCase, and I study the reaction that it catalyzes, and remember, ATCase does not make CTP. Remember, it's making this aspartyl carbamate molecule. All right, CTP is only made way down the line. That's not made by the ATCase enzyme. So if I take and I study the reaction that ATCase is catalyzing, and I study it in the presence of increasing concentrations of CTP, what I see is that the rate of formation, this is the product, we're seeing that the rate of formation is falling, and this is a log scale, so it's a fairly significant drop. This rate is falling as the CTP concentration increases. This graph is showing you visually what I've told you in words. The more CTP there is in the cell, the more the enzyme will be inhibited, but as I noted, we don't have a complete off switch. We're turning the volume way down, but we haven't turned it off. Okay. Now, interestingly, if we examine the catalytic activity, and this is not a very good representation of this enzyme, we discover something else very interesting. Okay? Now, the last figure I showed you showed the effect of increasing concentration of CTP. CTP is not a substrate. Remember, CTP is a product of that 10 steps away pathway. It's simply a molecule that binds to ATCase. So it's not a substrate for ATCase. If I take one of the substrates of ATCase and I measure that same rate of formation of product, I see a sigmoidal plot. This is the thing that we referred to on the last exam. That sigmoidal plot is happening because something else is going on in this enzyme. You saw the effect that CTP had. Now you see that a substrate is also having an effect. And how do I know it's having an effect? Well, I see a sigmoidal plot. It's not a very good S, but that's actually a sigmoidal plot. It tells us that not only can CTP affect the enzyme's activity, but so too can aspartic acid, one of the substrates. So a substrate can affect this enzyme's activity. We see at this point two regulators of the enzyme. This regulator is doing to ATCase very similar thing to what oxygen was doing to hemoglobin. The more of it there was, the more activity we see. And that means that aspartate is activating the enzyme. So the substrate, in this case, is activating the enzyme. Now, this actually has biological significance, and I want you to think, I'm going to tell you a little bit about that biological significance right now. Cells have to make nucleotides in order to make nucleic acid. They need to make RNA, they need to make protein, they need to make nucleotides. If a cell is getting ready to divide, the cell darn sure better be able to make enough nucleotides. If it doesn't have enough raw materials to make nucleotides, then that cell should not be preparing to divide, just like if your bank account is broken, you should not be buying beer for a party on Friday. Right? It's not a good career move. You may really regret it on Saturday, and I can guarantee you the cell will really regret it if it tries to go through the division process without having the resources it needs to make the nucleotides for the RNA and DNA necessary to do replication. Well, how does the cell tell if it's got enough? One of the ways is right here. If the cell has a lot of aspartate, what happens to the activity of this enzyme? It goes up. And what is aspartate? Aspartate is an essential building block not only for nucleotides, but also for proteins. And cells need proteins to divide as well. When the aspartate concentration is high, this is one of the signals to the cell that, hey, we can go ahead and have some whoopee. We can divide. That's cellular whoopee, by the way. So we can divide. You guys are slow today. Okay? So by actually having a little barometer, which is what this is, on the cellular nutrients, the cell is able to make an intelligent decision about to divide or not to divide. That's really cool. So not only are we regulating an enzyme, we're also testing the water. Is this stuff, do I have enough materials to go ahead and divide? Yes, sir. Is it only making this decision based on aspartate concentration? The answer is no, it's not. But this is one very useful piece of information. If cells didn't have enough aspartate, then you can see what would happen to this enzyme. The enzyme wouldn't go and it would stop everything else. So it's a good no switch. It's not the only yes switch. But a very good question. Okay.
If they don't have enough aspartate,
Yeah.
how is aspartate made?
How is aspartate made? Well, aspartate can be made by several mechanisms. There are metabolic pathways that can produce it. I'll mention a couple briefly next term. And they can also, cells can ingest it. So if they're floating around, you know, in a medium that's rich in amino acids or rich in proteins, they have sources of aspartate. Okay? Okay. So that's really cool. Now, what did I want to say here? I don't want to say anything there. I want to say a little bit of interesting things about the enzyme itself. Now, I'm focusing now on the protein structure of this enzyme. And when we study the protein structure of this enzyme, something interesting happens. Under certain conditions, and what we're looking at here is an analysis, a centrifugal analysis of this ATCase enzyme. It turns out ATCase has 12 subunits to it. So it's even more complicated than hemoglobin. It has 12 subunits. And if I am careful in how I manipulate those subunits, I can take that 12-unit piece and discover that it's composed of 3 pieces of an R2 dimer and 2 pieces of a C3 dimer. I'll show you what those are in a second. Okay? Now, in terms of the appearance of this protein, this is what it looks like. Okay? Now, we can exclude all the ribbons. I want you to focus on here. You see that the enzyme consists of 6 units called C, or catalytic. These are subunits where reactions are catalyzed. And it has 6 subunits that are called R, or regulatory. Now, you can't see all 6 of the catalytic because they're underneath. So here's the top 3, and then there's 3 underneath there as well. So we look at it from the side, there's 2 of the 3, and there's 2 of the 3 there. So this is like a double-decker of trimers right here. These are the catalytic subunits. And the regulatory subunits, you see 3 pairs of them here. Here's a pair, here's a pair, here's a pair. 3 sets of pairs of the regulatory subunits, and they are sort of hugging those catalytic subunits. Now, what we're going to see is that the allosteric effectors are going to change how these guys are all arranged. The allosteric effectors, in this case aspartate or CTP, are going to change the way that the regulatory subunits arrange themselves around the catalytic subunits. And these changes in structure will affect the catalytic activity. And just as we've talked about R and T state with hemoglobin, so too do we talk about R and T state with an enzyme. When the enzyme is in the very low activity state, it's in what we call the T state, or the tight state. When it's in the high activity state, it's in what we call the R state, or relaxed. Okay, so I'll show you some more figures of that in just a second. Okay. Now, before I do that, I need to introduce what will seem to you at first like a sort of a curveball. The curveball is, I want to spend a few minutes talking about an artificial substrate. An artificial substrate. It's in fact a suicide substrate, and though your book doesn't call it that, that's what it is. It's a suicide substrate. So what's a suicide molecule? What was the definition of that before? So it resembles a natural substrate. The enzyme binds it and it becomes covalently linked to it, right? Okay, so this molecule I'm getting ready to describe to you is a man-made molecule. It's not a natural substrate. It's something that we've made to study an enzyme. It resembles the natural substrate. It's not unlike aspartic acid. Not unlike aspartic acid. Okay? Here's what it looks like. All right? And we can see that when this guy comes in to the enzyme, it gets bound covalently to it, and here, I'm sorry, this is the synthesis of the molecule. Here is the artificial substrate. This artificial substrate will covalently link to the enzyme itself. Okay? It's called PALA, P-A-L-A, and I don't even know the name of it myself, so I always think of it as PALA. Well, what's the significance of that? Why do I tell you about that? Well, it turns out that if I take the enzyme, ATCase, and I add PALA to it, PALA binds as I expected before, but something unexpected happens. Study of enzyme linked to PALA first indicated that this enzyme could have 2 states. It could have a T state and an R state. When they take enzyme all by itself and you study it in a centrifuge, you basically see about 1 form. You don't see 2 forms. But when you treat it with PALA, you discover that the ones that haven't bound to PALA have 1 form, but the forms that have bound to PALA have a very different form. These correspond to the T state on the left and the R state on the right. Now you're sitting here very confused. You said our state was high activity, but this is a suicide inhibitor. If this is suicide inhibitor, it has no activity, right? That's correct. It turns out that what Paula does is it's catching the enzyme in that high activity state and freezing it there. Now, why didn't people see this before? The reason people didn't see this before is when the enzyme binds to the normal substrate, it catalyzes the reaction. It flips into R, it catalyzes the reaction, it flips back out, and you don't see it. Didn't see that state. There's about a factor of about, I think it's about 80 or actually a couple hundred to 1 that the T state is favored. So you don't even see that very tiny percentage that's the R state unless you lock things in it. And when they lock things in it with Paula, they discovered, wow, the R state exists for this enzyme. Now, if you look at what this R state looks like, you can see it's very different than the T state. In the R state, the enzyme is relaxed, it's opened up. Access to the catalytic units for the normal substrate is high. A normal substrate, if Paula weren't here, could get in here very easily and cause the enzyme to be able to bind it very readily. On the other hand, the T state, we see everything is uptight, it's up close. It's much more difficult for substrate to get into the catalytic subunit. And that's why we see the T state having less activity than the R state. So the T state, R state, very, very different states of these two enzymes. Paula allows us to see that. Okay. You'll notice the arrows here indicating that the T state is way to the left, the R state is way to the right. And you also notice something else on this screen, and that is the effect of CTP. Based on what you know about activities of enzymes and the allosteric inhibitors I've described to you, it's not surprising to think then that CTP, which reduces the enzymatic activity, is going to favor the T state, whereas aspartic acid is going to favor the R state. Now, what's of interest here is that they bind to different places on the protein. R stands for regulatory subunit. The regulatory subunit is where a regulator would bind, like CTP. And you might say, oh, well, then that means aspartate must bind the regulator as well, and it turns out it doesn't. Why? Because aspartate is a substrate. And substrates bind at the catalytic subunit, where the catalysis will occur. So aspartic acid binds to the catalytic subunit, and CTP binds to the regulatory subunit. Okay. Now, there's CTP locking it in the T state, and then we basically see that there. And there's the kinetic effect. If we measure the reaction in the presence or absence of CTP, you've seen this earlier, that you see a reduced activity. There it is in the presence of CTP. There it is in the absence of CTP. And so, not surprisingly, CTP is turning down that activity. Now, the last thing I want to say about this is something that is even more interesting. So this enzyme is playing a critical role in regulating CTP and in measuring the barometer of do we have enough nutrients, in this case aspartic acid, to go through and do replication. It turns out the enzyme responds to something else yet. All right? Well, let's think about this before I describe it to you. We think about that when we go to make RNA or we go to make DNA, if we have a lot of pyrimidines, okay, then we need purines, right? Because C pairs with G, G is a purine, C is a pyrimidine. T pairs with A, T is a pyrimidine, A is a purine. Okay? We want to have the right balance of nucleotides present in the cell. I told you if we make too much CTP, we've got trouble. But what if we have too many purines? What if we have a high level of ATP and GTP? What's going to happen? Well, we may have the same problem with mutation, and if we have that happen, one of the things we would like to do would be to increase the amount of pyrimidines, right? Because if we have a high level of purines and we raise pyrimidines, we're going to have roughly the same balance. And so it turns out that cells, that ATCase has the ability to sense this as well. Okay? ATCase is allosterically activated by ATP. It's allosterically activated by ATP. Here's the same curve you saw before, now in the presence of ATP, and you see we get increased activity in the presence of ATP. This serves as yet another barometer for the cell. So remember I said that the cell, ATCase is telling the cell, do you have enough amino acids in the form of aspartic acid to go through with division? ATP is an indication of a cell's energy. High ATP, high energy. If the cell is full of energy, the cell is full of purine nucleotides, and the cell has plenty of aspartic acid, this is a sign it's prime time to divide. Let's start making some pyrimidines. And that's what this is doing. So this enzyme is performing some very, very important functions in terms of helping cells to make intelligent decisions about dividing. ATP is an allosteric activator of the enzyme. And like CTP, it binds to the binds to the regulatory subunits. It binds to the regulatory subunits. That causes the enzyme to shift from T to R. So ATP is going to favor the R state. Aspartic acid is going to favor the R state. And CTP is going to favor the T state. Now, this is one of this really interesting control system that we see here is one of the reasons why ATCase has been one of the most studied enzymes in biochemistry. It's a classic enzyme for understanding allosteric regulation. And it's not the only enzyme that responds to more than one thing. This ability to respond to different molecules in different ways is really key to having elaborate controls over metabolic pathways. Very, very important. Okay, now I'm doing a lot of talking here. Let me ask for questions, and then I'll finish with a couple of things. Shannon?
So how is it that ATP indicates purine concentration?
ATP is a purine. So ATP is a purine, so when ATP is high, purine concentration is high. Yeah. Yes? Yes?
Causes the molecule not to shift, but causes it to stay in the T state, right?
Yes.
Whereas when ATP binds in R state or an R unit, it causes it to shift to the R configuration.
Okay, so his question is actually leading to my very next topic. So let me, it's a very good question. His question is, do CTP and ATP cause these changes, or is there something else that's involved? And I know that's not exactly what you asked, but that's what the implications of the question are. It turns out that neither one causes this to happen. So we thought of cause and effect with hemoglobin. We thought of the first oxygen caused the second one to be favored, caused the third one to be favored, caused the fourth one to be favored. So we saw cooperativity. And we talked about that as a cause and effect. Oxygen caused that to happen. Right? Well, there's other models that are consistent with changes that are not cause and effect. And that's what I'm getting ready to tell you about. Actually, maybe we'll save that for next time. But then I have two things for you. All right? So let me save the answer to that question for next time. I'm running out of time. One, I thought we would sing a song. And then two, I'll make an announcement about the exam. That way you'll sing loud. This is a song about taking exams. The term is almost at an end, 10 weeks since it began. I worried how my grade was because I did not have a plan. The first exam went not so well, I got a 63. 'Twas just about the average score in biochemistry. I buckled down the second time, did not sow my wild oats. I downloaded the videos and took a ton of notes. I learned about free energy and delta G naught prime. My score increased by 7 points, a C+ grade was mine. I sang the songs I memorized, I played the MP3s, I learned the citrate cycle, and I counted ATPs. I had electron transport down in all of complex V. I gasped when I saw my exam, it was a 93. So heading to the final stretch, I crammed my memory and came to class on sunny days for quizzing comedy. I packed a card with info and my brain almost burned out. 'Twas much to my delight I got the A I dreamed about. So here's the moral of the song, it doesn't pay to stew. If scores are not quite what you want and you don't have a clue, the answers get into your head when you know what to do. Watch videos, read highlights, and review. Okay. All right, now, the exams are graded. The average on the exam was 65.5. Not a bad one for the first exam. They are available for pickup in the BB office, ALS 2011. I just literally got them done just before I came to class. There's a key posted outside my office. You can look at the key outside my office, and I will post a grade distribution later this evening. High score on the exam, this is really interesting. The high score on the exam was 107, perfect, and the low was in the 20s. I want to say about 21.
I want to ask a quick question.
What's graded out as?
What do you mean what's the grade out? There's 107. Well, if you got the extra credit, you had 107.
Okay.
Right.
So if you got a 63, it's 63 out of 107.
You got a 63. A point is a point, right?
Yeah.
Right. So you got a 63 if you got a 63.
What percentage does that, like what grade in the class is that related to?
Well, I'll show the grades later.
Oh, okay.
Right?
That's what I did.
Oh no. This one.
Yeah.
R state or T state?
Our state.
Thank you.
Incredibly surprised.
Oops, excuse me.